Hello everyone. I'm very happy to share this work that we have done recently. I've never done video like this before. I thought it would be a good opportunity to show a little bit about what we do and why this is interesting. So today I'll talk about our recent work that is just accepted in Advanced Materials. So in this manuscript, we tried to present a hyaluronic acid cross-linked hydrogel that we use to print with the finest needle possible. And we can show in this paper that we can inject cells and we can make in vitro models. So this work was mainly done by a PhD student of mine. Her name is Shima, and she will be presenting to you in a few seconds. And this project was funded from Marie Curie project called Carthago, and I'm grateful for the European Union for supporting our work. Thank you very much.
Hello everyone. I want to talk about a little bit about the science of the project that Oommen mentioned. So in our initial work, which is published in Advanced Functional Material, we explored the cross-linking of hyaluronic acid with thiazolidine and disulfide, and there we could 3D bioprint human mesenchymal stem cells. But after that, we wanted to use only single compound of HA cross-linked with disulfide to use it for 3D bioprinting with very, very fine needle sizes. So that's why we tuned the gelation kinetics of disulfide formation by addition of potassium iodide. So the main novelty of this work is the addition of potassium iodide and accelerate the gelation kinetic at room temperature, because this hasn't been done before. And also usually something that has been well exposed is hydrogen peroxide, which in this work we substitute that with potassium iodide, that not only accelerates gelation kinetic but also has anti-inflammatory properties. So here I will show you some slides on 3D printed structures, because with the use of these small needles we could also build up and make really larger and more complicated structures that later on we use them also to produce in vitro 3D models to study different diseases. And here as an example I will present an osteoarthritis model that we made in the lab in vitro, and there we could study the interaction of human mesenchymal stem cells and also inflamed chondrocytes to see how they can affect each other in terms of their migration and also reduction of inflammation. This hydrogel allows us making like really porous and thin structures, but at the same time we can also print larger structures and more complicated. So here you can see that we made different structures with different geometries, and also we made a simplified ear structure using this hydrogel. And after that, we moved on to producing in vitro 3D models for different diseases. In this case, we made an osteoarthritis in vitro model where we could study the migration of the cells, in this case human mesenchymal stem cells and chondrocytes, and also the effect of MSCs on the inflammation in the osteoarthritis disease. So potentially this model in the future can be used to study the disease by itself, and also it's a platform to test different therapeutics. So I hope you like this work, and we are open for collaboration. So if you are interested to collaborate with us, please contact us. Our contact details and the link to the paper is also here in this video.